weekend? Anybody remember it? Or did it just go, it's gone, right? You wake up and all of a sudden the weekend's gone. I can't quite get the volume what I want. Can you hear me up there okay? Okay. I've been pleased. I've been talking with quite a few of you that are working the buffer problems, and that's a good sign. In my experience, getting started early is an important component, and making sure that you understand those is important as well. They have different levels of complexity, and I will tell you that when you get to the one that has the exceeding a buffer capacity, I will not expect you to calculate the pH after you exceed the buffer's capacity. That's been an anxiety for a few students. I'm not going to do that. The problem is there mainly for you to recognize when you have exceeded a buffer's capacity. Okay? So the TAs are going through the problems in the recitations, and hopefully that is helpful. Is that helping or not helping, or what's your experience? Helping? Okay, good. Not helping? Surely it's not unanimous. Nobody's going to say, right? Helping. Okay, good. All right. What I want to do today is dive more into protein structure. So I've said some things about amino acids. The TAs will be going through some calculation of charge problems for you. And I will also tell you that there are videos of me solving problems of protein and amino acid charge that are on the class website. In fact, I will just show you that since I've had a couple of questions. The videos for those are over here on the right side. They're different from the videos that are over here. So if you look over here, you'll see some videos of me working a bunch of problems on that. Okay, well, last time I got started talking about primary structure of protein, and I will remind you that when we talk about protein structure, we can think of it as occurring at 4 different levels: primary, secondary, tertiary, and quaternary. And so today I'm going to talk about primary, and I will also talk about secondary, and I don't know if I'll get into tertiary or not, but if I get through secondary today, I'll be happy. The primary structure of a protein is the sequence of amino acids comprising a protein. The sequence. And the sequence is absolutely essential. As I mentioned last time, the sequence determines all of the other structures. The primary, that is the sequence of the protein, the primary structure determines what the secondary structure will be, the tertiary structure will be, and the quaternary structure will be. All right? So that primary sequence of a protein is important. When cells have mutation, and that mutation affects a coding for a protein, the mutations that affect the sequence of the amino acids will be the most important ones, and will be the ones that have the most drastic effects. Okay? So that primary structure is very, very critical. Now, this shows on the screen a polypeptide. It is unfortunately not the best polypeptide they could have picked, but I guess for our purposes, for what we need at this point, it'll work okay. What we see is a polypeptide that has 5 amino acids in it. And we see the 2 ends that I talked about before. You remember that every polypeptide, and by the way, I use the term polypeptide and protein interchangeably. Okay? Technically, that's not right. All right? But it's a fine line of distinction, and I'm not going to make that distinction in this class. If I say polypeptide or protein, we will use those terms interchangeably. Okay? Now, as I said at the very end of the lecture last time, there are ends of a protein. There's an amino end, which always has a free alpha amino group. We can see this is the amino end over here because it has a free alpha amino group. And we see this is the carboxyl end over here because it has a free alpha carboxyl group. That's the only place in a polypeptide where we will see a free alpha amino and a free alpha carboxyl. And the reason for that is because the peptide bond, which you see right there, gobbles up a free carboxyl and a free amino. So every time we have a peptide bond, we lose a free alpha carboxyl and a free alpha amino. So we see peptide bond, peptide bond, peptide bond, peptide bond. Okay? Now, another thing that we see in this schematic is that this actually is a nice simplification of the structure of a protein. This is an R group, okay? This is an R group. 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 You notice the pattern. Up, down, up, okay? So we see alternating sides of this structure that the R groups are on. Now, that's not totally surprising. Some of the R groups are rather large, okay? Look at the size of the R group on tyrosine. Look at the size of the R group on phenylalanine. If we try to put them on the same side of the polypeptide, they're bulky. And we're going to run into problems with atoms that don't want to be close together. We've already seen the energy issue with that. Proteins arrange themselves by shifting bonds to keep those R groups as much as it can away from each other. So one of the ways it does it is based on what you see right here on the screen. As I will describe to you in just a little bit, that orientation gives us a configuration we refer to as Strands, and I will explain why that's the case in just a second. The other thing I want you to notice about this is that these, I guess I've said it, that there are peptide bonds that are joining each of the 5 amino acids together. Now, there's a better schematic that I'll show you in just a second that will depict this for you a little bit more clearly. I do want you to notice right here the alpha carboxyl group, okay? Alpha carbon group. The alpha carbon group right here is the one that has the R group on it, the R group on it, the R group. So the alpha carbon is going to turn out to be an interesting carbon in this overall structure. Now, schematically, what I showed you on the last figure is this right here. And I told you in words. There's the first R group, there's the second, there's the third, there's the fourth, and there's the fifth. And the R groups have arranged themselves so that they're pointing away from each other. Peptide bonds are interesting structures, okay? Peptide bonds are something that can form what's called a resonant structure. And you learned in organic chemistry that resonant structures arise as equivalent electronic configurations for certain atoms. The resonant structure of a peptide bond, this structure is essentially equivalent to this structure on the right. Well, this structure on the right, as we look at it, has a double bond. And what we learned about what we learned in organic chemistry about double bonds is the fact that there are some specific stereochemical orientations that can happen with those. Those specific orientations can create what we think of as cis bonds and what we think of as trans bonds. Okay? So here is an alpha carbon, here is an alpha carbon, and you will notice that they are oriented with respect to the double bond in a trans configuration. This one being up, this one being down. Okay? Now that turns out to be very, very important for understanding the overall structure of a protein. Okay? So even though that resonant structure isn't a double bond all of the time, it behaves as if it is almost all of the time. So this cis-trans nature of these alpha carbons are very, very important for us to understand protein structure. Okay? So I'll emphasize again that resonant structure gives rise to what are cis or trans. And by the way, cis or trans can exist. What we see when we analyze the structure of proteins is that the trans is very strongly favored. If the trans is not very strongly favored, we could imagine that when we have a cis, we would have this bond going down. And as this bond goes down, the R groups may get into each other's face. And that's exactly what happens in some cases. So having that structure as a trans is important. Okay? This now shows the peptide bond, as you see here, the carbon to nitrogen, as if it were a double bond. Well, what you remember from organic chemistry is that that double bond defines a plane. Double bonds don't rotate. Double bonds are fixed, and that forms a plane. The plane of that double bond is shown what you see on that blue in the screen. Okay? Again, we see the alpha carbon in the trans configuration relative to that double bond. Notice now, again, here the R group is sticking out. There's the R group sticking down. The R groups are oriented away from each other as much as possible. Okay. Now, this figure shows us what that structure will look like if we try to put that peptide bond into a cis configuration. If we try to put it into a cis configuration, now here's our big bulky R group, here's our bulky R group. Look, they may run into each other. It's for this reason that we see the trans configuration favored something like 99.999% of the time. Okay? At least 99.999% of the time, we see the trans double bond, or the trans configuration favored, not the cis configuration. We do occasionally see the cis. Okay? Now, one of the amino acids actually favors, at least relatively favors, the cis configuration. It's the amino acid called proline. And when I described the structures of the amino acids to you, I neglected to point out something very important about proline. Proline is the only amino acid whose R group makes a bond with the alpha amino. Okay, we can see that right here. Here's the alpha carbon, here is the R group coming off, and we see that the R group makes a bond with the alpha amino group. Okay? Now, the significance of that is because this is a bond to the alpha amino, there is less flexibility associated with a proline. Prolines do not have as much ability to rotate bonds as do the other amino acids. Further, we see proline has some things hanging off the end of it, okay? And those things hanging off of the end of it, in either case, can get in the way of a trans or a cis. So proline is an oddball as far as the amino acids go, and proline has a very strong effect on the structure of proteins in which it's found. It's not uncommon where we find a proline in a protein that we actually see something called a bend. Okay? Now, I'll explain bends to you in a little bit. But bends arise because proline is not very flexible and it has some real structural limitations, and the rest of the protein has to go along with whatever proline defines. Now, this favoring the cis is only relative. Okay? The trans for proline is still strongly favored. Probably 99% are still in the trans. But about 1% of the time, it'll flip into the cis. The other ones won't have that happen nearly so frequently. So even though proline is more relatively favored, it's still probably 99% of the time hits the, has the trans configuration set for it. I'll have a lot more to say about proline as we get going further along, talking about structures of proteins. Questions about that? Yeah.
So it only favors cis more than the other amino acids.
That's correct.
But it's still favored as trans.
It's still favored as trans. That's correct. Uh-huh. Yes, sir. The alpha carbon is on this guy right here. Okay? So we see the alpha carbon. Let's see. Yeah, the alpha carbon's right there. So we see that his R group is bending back over on this guy. Okay? See it? It's making that bond with the alpha amino group. That's the only amino acid that does that, and that causes a structural limitation on the overall protein at that point. Yes, sir?
Can you go back to the webpage that showed all the amino acids in the sequence of primary structure with the stereosymes?
Oh, yeah. You're talking about back here?
Yeah. So are all the alpha carbons in the same configuration as in terms of R and S?
Are they in the same configuration with respect to R and S?
Yeah.
I would have to sit down and do it. I don't know off the top of my head. Yes, Shannon?
So with cis and trans proline, it can rotate in order, so it is rotating for that, but it just isn't as flexible around the carbon nitrogen.
It's not as flexible around there. So it's rotating. Remember, we've got a double bond. It could be in the cis or trans. That's the rotation that we're talking about. And so the flexibility I'm talking about, it refers to the rest of the molecule. The cis and trans of the peptide bond are still are capable of flipping. Okay? So and as I will show you in just a little bit, with proline what happens is because that alpha carbon is in a ring, we don't have the flexibility of the alpha carbon bonds to rotate in the same way that we have in the other amino acids. Okay? Okay, where are we at here? All right, well, now after I've said something about that, I've told you that the alpha carbons are very important for us to understand something about protein structure. It's important now that we think about the alpha carbons in that overall scheme. Okay? Here's our peptide bond right here. Peptide bond is behaving as if it's a double bond. All right, there are 3 bonds that are of interest to us, however, in a given amino acid. Here's a bond between the alpha amine and the alpha carbon. Here's a bond between the alpha carbon and the alpha carboxyl. All right? Only the peptide bond is capable of being a double bond. These guys are each capable of, they're perfectly single bonds. And single bonds, you recall, can rotate. Okay? Rotation is very, very important for the overall structure of a protein because rotation gives enormous possible structures that can arise. All right? Now, I'm going to introduce a concept that I want you to have a general understanding of, but I'm not going to go into the specifics of the actual angles. Somebody's already asked me, what's the 0 point for the angles? And that's not really what's important for us. Okay? Because we have the ability to rotate across these 2 double bonds, we could imagine that the structure of this protein will partly be a function of how those rotational angles are in fact set up. Okay? Let's think about this. This guy right here is part of a plane, right? We can think about this guy as being the plane of one peptide bond. All right? So here's my peptide bond on the left. On the right, I've got another peptide bond. It's also a plane. Okay? Everybody with me? Peptide bond on the left, peptide bond on the right. Okay, when I put my thumbs together, the place where my thumbs are make that alpha carbon. Okay, when I pull this up, it rings. All right? The alpha carbon's in between my thumbs. All right? Now, what happens is that there's rotation that's possible. Those planes themselves can rotate around that alpha carbon. And now we start thinking, oh, wow, these rotations can, in fact, also have limitations in terms of the things that are out here. The things that are out here may start bumping into each other. So there's going to be some limits on the way that this guy can rotate and the way that this guy can rotate. All right? Those 2 bonds are called phi and psi. And phi and psi specifically are rotational angles around the alpha carbon. Okay? Phi is between the alpha amine and the alpha carbon. Psi is between the alpha carbon and the alpha carboxyl. Phi and psi. So you'll hear a lot when you talk about protein structure with respect to what phi and psi actually are. Everybody understand phi and psi? Now, keep in mind, they are rotational angles. We're not talking about this kind of angle. We're talking about rotation. Rotational angles have some very, as we will see, some very strict limits on them because of the spatial consideration that we've talked about before. Those spatial considerations cause major limits for what things are actually stable. Now, there's a famous Indian scientist named Ramachandran. You don't need to know the name, but Ramachandran was very astute in recognizing the importance of these phi and psi angles, because he recognized that those were the primary variables in determining certain structures of proteins. And so he plugged it into a computer, and he plugs into a computer and says, here's the geometry of the peptide that I've got. Here are these groups that are floating out here in space. Okay? Where are things going to be too close together? Because if I know if I get too close together, the energy gets, whoa, prohibitive. Right? So he plugs into a computer and just starts rotating through space and determining where the angles are that are stable, that is, they have relatively low energy, they have plenty of room, and where are the angles that are very unstable, where they have high energy and would come apart. So he created something we call a Ramachandran plot. Ramachandran plots plot the angles of phi and psi. Now, I'm just showing you one. I don't want you to panic with this. Okay? This is mostly informational. I'm not going to ask you to interpret a Ramachandran plot. Okay? However, Ramachandran plots are very interesting because what we see when we look at a Ramachandran plot, what you see is exactly that. You see on the y-axis psi through 360 degrees of rotation from plus 180 to minus 180. For our purposes, the moment doesn't matter. In fact, it doesn't matter at all for our purposes where 0 is. It's an arbitrary starting point for us right now. Similarly, phi along the x-axis goes from minus 180 to plus 180. Right? So he asked the computer, tell me where the regions are that will be the most stable, that will be the lowest energy, not the most ones that are inhibited. And what he found was that there were two major regions that were there. One was right here, and one was down here. Okay? No, you're not going to memorize those angles or any of that sort of stuff. But this is interesting. A good deal of that space that was out there, all right, that was possible for rotational angles, gave rise to structures that were not very stable. Okay? That meant that there was relatively limited amounts of angles that gave rise to stable structures, and they seem to be clustered pretty much into two regions. We'll see those two regions turn out to be very important for our understanding of the next level of protein structure. Okay. Everybody with me on this? Questions about Ramachandran plots? I'm going to say a little bit more about them in just a little bit also. Okay. Well, that's what I want to say about primary structure. And when I'm talking Ramachandran plots, as we will see, we're starting to talk about the next level of protein structure. That's called secondary structure. Now, secondary structure, I'm going to give you a definition. Secondary structure is the next higher level of protein structure, and it arises as a result of interactions between amino acids that are relatively close in primary sequence. I'll repeat that. All right? Secondary structure arises as a result of interactions between amino acids that are close in primary sequence. They're close interactions. We don't see things very far away interacting in secondary structure. Now, technically, secondary structure involves a regular repeating structure as well. I didn't include that in the definition, but technically it does mean it's a regular repeating structure. I'm going to show you some regular repeating structures in just a bit. Okay? The regular repeating structures I'm going to show you arise because of those limitations that we saw in the Ramachandran plot. Okay? Well, let's think about this. Here is a regular repeating structure we commonly find in proteins. It's one of the structures for which Linus Pauling was recognized ultimately with a Nobel Prize. Okay? It's called the alpha helix. The alpha helix, as you can see by the structure on the screen, is a regular repeating structure. It's a coil that goes on and on. You've seen DNA. Everybody's seen DNA. But DNA is a double helix. This is a single helix. Okay? Now, this shows three different views of an alpha helix. And from this perspective of the alpha helix, we can see certainly here the helical nature of it here. It's not quite so easy to see the helical nature of it here. But what we see is that there are some hydrogen bonds. See those green dots right there, or the green dashes? Those are hydrogen bonds that are helping to stabilize the secondary this structure for an alpha helix. Okay? So hydrogen bonds are stabilizing this structure. Very important point. The most important bonds stabilizing secondary structure are hydrogen bonds. And they're happening within a few amino acids of each other. Now, we could go through and we could do all the business of how many amino acids they are apart and how many there are per turn and so forth. And I don't think that really tells us anything that's important about this structure. Okay? The most important things about this structure are the regularity, the hydrogen bonds, and the last thing I'm going to mention, which is this thing right here. If we notice in that third panel in C, we can look sort of down the barrel of the alpha helix, and when we do, look where the green groups all arise. How have they been arranged, guys? Outside the helix, right? Again, we come back to this important point about bulky molecules need their space. R groups are oriented in an alpha helix to the outside. Okay? Now, one of the things that we will see about alpha helices is that they are parts of the overall structure of proteins. Some proteins have almost exclusively alpha helix, and that's all they have. They just go on and on and on, kind of like the ever-ready bunny. Okay? Ha-ha. All right? In other cases, more commonly we see that they will go on for a ways, and then we see another structure arise, etc. If I have a protein that really only has alpha helix, I have something called a fibrous protein. Fibrous protein. A fibrous protein has primary structure, it has secondary structure, but that's primarily about all it has. And it goes on and on and on and on. Example, my hair. Okay? Hair, okay, has keratin that has a structure that just goes on and on and on and on. Okay? It's fairly boring as proteins go. We'll see some much more interesting proteins than that. But fibrous proteins have that characteristic. Okay. Now, this shows you the orientation, and it's showing you on that schematic figure that you saw before where the hydrogen bonds are located. Notice that this hydrogen bond that is forming with this amino acid that's several amino acids away from it, this hydrogen bond couldn't interact like this unless there were a coil. It's the coil that allows the hydrogen bonds to form, and conversely, it's the hydrogen bonds that help to stabilize the coil. There's a carbonyl, there's a hydrogen. And as we saw on the very first day, those are really good pairs for making hydrogen bonds. Okay. If we go back to our Ramachandran plot, what do we see? There is where the alpha helix is found. Okay? The alpha helix, when we look at all the alpha helices that are out there, all right, we see that all the alpha helices map in this region very, very tightly. And there's a lot of things with very similar angles to alpha helix, out here, for example, that have Ramachandran angles very much like it. The alpha helix is in a very, very stable region of the Ramachandran plot. That's not surprising. Not surprising at all. Okay? Make sense? I'm going on and on and on. You guys want a joke?
Sure.
It's a little dull in here, right? Okay. So this is one of my favorite jokes, all right? So there's this little guy named Artie, right? And he wants to be a hitman. His dream is that he can go and he can kill people for a living, right? Make a lot of money in this, right? He's got a career set for him in the mafia or something, right? He decides to go out and do this. So he figures, well, I got to get started. So he goes out. He makes a little note, he tacks it up on the bulletin boards around town. He tacks it up on the telephone poles and so on and so forth. And says, we'll kill someone for cheap, right? We'll kill someone for cheap. And so he's got his thing all up, and this guy calls up and says, yeah, he says, got somebody I want you to kill. He says, oh yeah? He says, yeah, he says, I'd like you to kill my wife. And he says, okay. He says, how you want me to kill her? And he says, I want you to strangle her. No problem. He writes this all down. And, where might I find her? He says, well, as a matter of fact, she's at the grocery store right now. He says, okay. He says, can you kill her right now? And he said, yeah. And he said, how much would you charge? And Artie says, well, I'm getting started. He says, I'll do it for $1. Okay. That's how you get started, folks, He says, I'll do it for $1. The guy says, that's great, yeah. So Artie trots off to the grocery store. He gets out there and he looks, and she's right there in the middle of the produce section. He looks around, you know, and there's nobody there. He goes up and he grabs her by the throat, strangles her right there in the produce section. Yes, I'm set. Uh-oh, somebody saw him, okay? Somebody saw him, right? Damn, this could be all screwed up. So he goes, he grabs this person, he strangles them. Can't have a witness, right? I mean, if you're going to get started, you can't have witnesses. So he goes and he strangles this person right there in the grocery store. He's all, uh-oh. There's a third one. Right? He goes over, my lucky day, right? So this wasn't the way I envisioned this would get started. So he goes over, he grabs this person, he strangles them, he looks around, and he goes racing out of the grocery store. And the police catch him, and the next day the headline in the newspaper says, artichokes 3 for $1 at the grocery store. Oh, that was bad, wasn't it? Artichokes 3 for, okay. Okay, I will tell you some jokes later in the term, and so I want you to remember, okay, that all I have to do is say artichokes 3 for $1 at the grocery store, and you're going to laugh at those. I mean, you may not laugh at them, but if you do, then there's my punchline that works. So, okay, artichokes. Here's an alpha helix. Here's a schematic representation of the structure of a protein showing alpha helices. You notice that this is not a fibrous protein. This is a protein that has an alpha helix that goes for a little ways, and then we have a bend. And then it goes for a ways, and then we have a bend. And then it goes for a ways, and then we have a bend, etc. Okay? What we see about the structure of most proteins is that they have regular repeating structures for a certain region, and then something kind of interferes with their ability to remain alpha helical in nature. I've mentioned one amino acid today that might interfere with that. What would you suppose that would be? Proline, okay? Proline is going to have some limitations in terms of angles, and proline may, in fact, interfere with the regular helical nature that we see here. Now, I'm going to show you an exception, actually, probably on Wednesday to that, but proline is one that can really interfere with a regular repeating structure. Okay. A second structure that is a repeating structure that is, in fact, a secondary structure is known as a beta strand. Okay? Let's first of all look where beta strands appear. Beta strands appear in Ramachandran plots, as you can see here. No surprise. Again, in the most stable region of a protein. You can see it's actually a bigger stable region of the Ramachandran plot. Yeah? I can't see. Where are you at? There you go. Okay. Yes. There are right-handed and left-handed, but right-handed is by far the predominant. In fact, some people argue if left-handed even occurs. Yeah. But right-handed is the predominant form, yes. Was that the question, or was there something else? No. Okay, so what you'll see is that the orientation actually, if you want to come by my office, I'll show you an example of a right-handed versus a left-handed helix. And they do differ. And if you flip it upside down, it still remains a right-handed helix. It has nothing to do with the orientation. But come by, I'll show you, okay? Good question. Okay. Beta strands. Beta strands, and this is a little harder to see in this image. I'm going to show you actually a better image of that. It's going to look very much like that first one that I showed you, which was the up, down, right? That very first image where I showed you the protein where the R groups were oriented up, down with respect to each other is a very good model for what we call beta strands. And I call them beta strands because there is a strand. When I put them together in a bunch of strands, I make something called a sheet. Okay? People commonly call beta strands beta sheets frequently because they are arranged in sheets. Silk, for example, is composed of beta sheets. All right? Now, look at the orientation here of the R groups. In this case, they're going out of the plane of the board, in, out, in. That way versus this way, that way versus this way. And they're alternating as they are set up here, okay? They are arranged so as to, again, space those groups out so that they're not causing problems energetically due to their close interactions with each other. Now, these are what are described as anti-parallel, and these here are described as parallel. For our purposes, it doesn't really matter, but if you're curious, I will tell you, okay? This would be an example. This one is parallel. That would mean we're going from alpha to carboxyl and alpha to carboxyl in the same direction. Whereas if they twist around like this, they're what's called anti-parallel. All right? That's all that means. And strands can be twisted and turned, bent as appropriate to form the structures necessary. Gesundheit. Here is an example of a protein that has beta sheets, and they're arranged in the form of a barrel. We see barrel structures arising in proteins to help perform important functions. I'll talk about a couple of them later in the term. But these barrels are just literally like a barrel is. Okay? So we see at the nanoscopic level that structures that we can recognize on a macroscopic level in the real world. Yes?
Are there any beta sheets that have parallel and anti-parallel sections?
Are there beta sheets that have parallel and anti-parallel? I'm sure there are. I couldn't name one for you off the top of my head, but yes, yes. So they're not exclusive to one way or the other. Okay. Now, I mentioned turns earlier today. All right? Turns are very important because it's turns that interrupt secondary structure. All right? Turns interrupt secondary structure. And when we see a turn, there's a variety of configurations they can have, but a common form has been identified that involves, in this case, 4 different amino acids. There are some that involve 3, there are some that involve even more. But suffice it to say that this is a common structure that we see. All right? Not surprisingly, one of these amino acids is proline. Very commonly, proline. Now, something that may surprise you is another one of the amino acids that's involved in this structure, commonly, and again, this is not absolute, but commonly, is glycine. Glycine is the amino acid that has the smallest R group. It only has a hydrogen. And you would say, well, why would glycine be involved in a turn? What do you think? There's just mumbling. What's that? Not because it's achiral. No, it's achiral because it has the small R group, but it's related to the small R group. Yeah?
Is it a reduced steric hindrance?
There's reduced steric hindrance. Glycine allows for a lot more flexibility. Glycine allows for a lot more things to happen. So it actually is favored because we've got this limitation over here and we've got flexibility over here. Now we may, in fact, be able to do something that we couldn't do otherwise, and glycine will facilitate that. Again, it's not absolute, but we do commonly see that in turns. Okay. And I won't go through that. Okay, now, I mentioned with respect to alpha helices that we see them in what are called fibrous proteins. We also see them in beta strands. As I said, silk is a protein that's comprised of beta sheets, that is a bunch of strands put together. Silk is also a fibrous protein. Your nails, your fingernails are fibrous proteins, okay? So fibrous proteins are comprised, as I said earlier, of primary structure and secondary structure, but they have very little tertiary or quaternary structure, as I will be talking about later. Okay? So here is an example of a fibrous protein. We see that in this case we have helical structures, and the helical structures themselves are intertwined. This gives rise, as we will see in some cases, to strength of structures. And I'll have an example for you again next time. Okay. This shows a very interesting characteristic of protein or portions of a protein. These can be separate proteins, or these can be portions of the same protein that are interacting with each other. Now, I'd like you to look at what's going on with this. When people first discovered these structures, they found something very interesting as they were examining amino acid sequences of proteins. By the way, when we find a new protein, we always want to determine its amino acid sequence. Okay? Because as we know, the amino acid sequence gives rise to everything else. And we know the amino acid sequence long before we know the overall structure of a protein. We can make some predictions, but our predictions, as I will tell you later, aren't as good as we would like them to be. So the first thing they noticed about this class of proteins when they discovered it was that it had a very interesting thing in its primary structure. Every 7 amino acids or so, there was a leucine. Okay? And that was a little bit of a puzzle. Why is there every 7 amino acids a leucine? And when they started determining structures, it all made sense. Leucine is one of the hydrophobic amino acids. It has a hydrophobic side chain. That hydrophobic side chain, as you recall, doesn't like water. When they examined this, here's one strand with its every 7 amino acids we see a leucine. Here's another strand, and say either part of the same protein or part of another protein, that every 7 amino acids also has a leucine, and look what they are doing. They're interacting with each other. They're getting away from water by interacting with each other, and they're forming a structure that we call a leucine zipper. This leucine zipper arises because of the regular nature of the alpha helix. That regular repeating structure is placing that leucine at the same place out there in space every time, allowing those leucines to interact. And so we could imagine that if we wanted to peel these apart, we would do it just like a zipper. And that peeling apart is going to be relatively easy to do because these are only hydrophobic interactions that are actually helping to hold these leucines together. We'll talk a little bit more about that in a bit. Okay? So leucine zippers arise because of the regularity of the alpha helix. Okay? Now, the structure that you see on the screen actually is not a secondary structure. The alpha helix is a secondary structure, but now we're starting to see what we refer to as tertiary or quaternary structure. In this case, if it's in the same protein, it's tertiary structure. Now, I'm going to show you some other examples of that in a second. But when we see different regions of proteins that are not close to each other interacting, we see what we call tertiary structure. The leucine zipper can be a part of a tertiary structure. That's correct. Okay? I said I was going to talk about it next time, but I guess I'll talk about it today. I told you I had an exception to the rule about proline forming structures that are helical in nature. All right? One of the most important fibrous proteins in nature is the most abundant protein in your body. It's known as collagen. Collagen is literally the glue that sticks you together. It holds us together. Without collagen, we are in trouble. Okay? Collagen, to give you an idea, has a structure that looks something like this. Coils of coils like we saw before. Okay? Those coils of coils, okay, when we analyze the sequence of amino acids comprising them, we discover something very surprising. Look at this sequence. Look at every place where you see proline, 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 proline. It's full of proline. Yet we have a regular repeating structure. You might wonder, what's that HYP? HYP stands for hydroxyproline. So not only do we have proline, we have a modified form of proline here called hydroxyproline, and this guy is just bursting with this stuff. Yet it forms a regular repeating structure. How is that possible? Well, the answer is in red on the screen. Again, glycine is there, and glycine is giving the space needed to form this regular repeating structure. Essentially, everywhere we see a proline, we see a glycine. Okay? This is facilitating formation of that regular repeating structure. Okay? So this is one of our exceptions. As I said, proline is not an absolute thing for a term. But there's an interesting story that goes with this, and it's that story that I'll finish with today. Okay? Hydroxyproline is a modified form of proline. When I told you about the 20 amino acids, I said there were amino acids that got modified after they were made in a protein. And hydroxyproline is an example of one of those amino acids. It's put into the protein as proline, but then it gets modified, okay, chemically. That chemical modification involves putting a hydroxyl group on it. And that hydroxyl group ultimately comes from vitamin C. One of the few reactions where we actually have a vitamin that's playing an important role in a chemical process. Vitamin C is ultimately the source of this. We make hydroxyproline, and one of the reasons that we have to have hydroxyproline is so we can make strong collagen. Okay? Now, this was discovered partly originally back in the days of the old pirates. The pirates would go out, big, honking, hairy, stinky, ugly guys going out, conquering the world, killing and robbing and doing all kinds of nasty things. And they would go out on the ocean for months at a time, and they ate salted meat because they didn't have refrigeration. They didn't exactly have arugula. They didn't have any fruit, desserts, okay? They had no source of vitamin C. And so they go out as these big, honking, ugly guys, and they come back as these puny little wimps. They develop a condition called scurvy. And that scurvy arises from vitamin C, and I'm going to tell you what is involved in scurvy. Okay? What's involved in scurvy? Well, these hydroxyl groups that we put onto proline are very important, all right? Because it turns out that these hydroxyl groups are reactive with each other, such that when we start putting them together, they'll make bonds with each other. We can actually tie these strands together with covalent bonds that arise from those hydroxyl groups on proline. If you ever braid your hair, you got to tie it off with something at the bottom, right? Otherwise, the braid falls out. These chemical bonds that form between the hydroxyls are actually linking those strands together and keeping them from falling apart and giving strength to the collagen. As a result of vitamin C, you have strong collagen. You don't fall apart. If you don't have vitamin C, you develop scurvy. You have weak collagen, and literally you fall apart. That's what happens with that. Okay. I was going to sing a song, but I think we'll call it a day and save that for another time. See you guys on Wednesday. How you doing?
Are there any other hydrophobic amino acids that form a zipper, or is it just leucine?
Leucine zippers are the best well-known ones.
Okay.
Definitely. Yeah, it's a good question. I should write one, I suppose, shouldn't I? All right.